Hi guys, in this video we are going to discuss about clinical physiology of R.B.C count in detail. Aim: to find out red blood cell count per cubic millimeter of the blood sample provided. Principle: blood is diluted 1:200 ratio with Hayem's fluid. Counting of R.B.C done in the definite volume of diluted blood. This count is multiple with the dilution factor and volume factor, so as to find the number of R.B.Cs in 1 cubic millimeter of undiluted blood. Apparatus required: Berger's counting chamber, R.B.C pipette with 0.5 and 101 marking, spirit, cotton, R.B.C diluting fluid, lancet. R.B.C diluting fluid and composition. Hayem's fluid. Composition: mercuric chloride 0.5 gram anticoagulant agent. Sodium chloride 1 gram maintain isotonicity. Sodium sulfate 5.0 gram. Prevent Rowley-Orr's formation. Distilled water 20 milliliters solvent. Procedure: dry and clean the pipette. Clean the counting chamber and cover slip free from grease. If the blood is not given, take the blood from the fingertip palmar aspect. The skin should be sterilized by alcohol and prick with lance. The prick should be sufficient deep to cause the bleeding without squeezing. Don't squeeze the tissue because the tissue fluid emerges out and mixes with the blood. It causes incorrect count of erythrocyte. If the sample blood is given, it should be shaken well before taking in the pipette. Hold the pipette in the horizontal position and draw the blood exactly up to mark 0.5. If the blood is past 0.5 marking, withdraw the blood from the pipette. Insert the tip of the pipette vertical into the diluting fluid and fill 101 mark just above the bulb. When it's filled, bring the pipette to a horizontally between the palms and roll gently for about a minute to ensure thorough mixing of the blood and fluid. This will give a suspension of R.B.Cs in the bulb of the pipette. Dilution, 1:200 ratio. Clean cover slip is placed in the Berger's chamber so as to cover the ruled R on both sides. Focus the R.B.C squares of Berger's chamber ruling under the low power objective of microscope. Discard the first few drops of fluid from the R.B.C pipette. They constitute the unmixed Hayem's fluid present in the stem of pipette. Allow some time for cells to settle down in the counting chamber, so that all cells present will be in the same plane. Check whether cells are uniformly distributed or not. If not, clean the Berger's chamber and recharge it. Focus the R.B.C squares under high power objective, 40x, and count R.B.C in 4 medium-size corner squares and 1 medium-size central square, 1x5 mm side, that is in 80 small squares. Rules of counting of cells. In order to count the cell twice, following needs to be observed. Any cell is in the upper left border of the square is counted. Omit the cell lying in the right lower border of the square. If the cell lying in the right upper border of the square, it is counted. If it is lying in the left or lower border of the square, it is omitted. In square bordered by triple line, the middle of the triple line is assumed to be the boundary of the square. Count the R.B.C in 4 medium-size corner of the square and 1 medium-size central square. Total number of erythrocyte is 447. Calculation: volume of 1 medium square equals L into B into H millimeter cube. Equals 1/5 into 1/5 into 1/10. Equals 1/250 millimeter. 1/250 cubic mm into 5 equals 1/50th millimeter cube. 1/50th millimeter cube of blood contain equals 447 no of cells. 1 millimeter cube of blood contains equals 447 into 50 into diluting factor. Equals 447 into 50 into 200. Equals 447. That is 1 millimeter cube of blood contain 4.47 million cells per millimeter cube. Normal R.B.C count. Males, 5 to 6 million cell per cubic millimeters. Female, 4 to 5 million cells per cubic millimeters. Inference. Total R.B.C count as with the normal limit. Result. Thus 1 cubic millimeter of blood contains 4.47 million cells per cubic millimeter. Thanks for watching. Dentology Notes. Please subscribe.